This video will cover the electron transport chain. We talked about the steps that precede the electron transport chain. So now we need to talk about the proteins in the electron transport chain. We discussed how they are embedded in the mitochondrial membrane and the fact that some bacteria and archaea have similar proteins. Of course, they don't have the mitochondria, but they have doubled up membranes or folded membranes to form a similar structure or layout for this system. Now, that's because the electron transport chain is extremely efficient in making ATP, as we could see from this excellent and very accurate meme. So the electron transport chain is responsible for producing most of the energy in a mammalian cell, in most eukaryotic cells. And glycolysis is responsible for making a relatively small amount of energy. Remember that the ATP that is produced during glycolysis is produced through substrate level phosphorylation. So more about the electron transport chain. There are many enzymes and they all work in sequence to perform the task of forming a proton gradient. The proton gradient is then later used by ATP synthase to bring together an inorganic phosphate group and an ADP molecule. We'll talk about that in another video. So let's have a quick summary of what we talked about in glycolysis and the following steps. So in glycolysis, we begin to break down glucose. We do not lose any carbons in glycolysis, but we do use 2 ATP and produce 4 ATP. So there's a net production of 2 ATP in glycolysis. Then there's pyruvate processing. There is no ATP production in pyruvate processing, although we do lose one of the carbons from each of the glucose molecules. So we lose 2 carbons from the original glucose molecule. And in the Krebs cycle, we continue to lose carbons through carbon dioxide. And in total, between all of these processes, we release all 6 carbons from a glucose if the process completes. We produce 10 NADH, 2 FADH2, and remember this is in part because we have 2 halves of the glucose molecule entering into the citric acid cycle, and we produce 4 ATP, but 2 net, and then 2 GTP also from the citric acid cycle. So where does the rest of the ATP come from? Because we normally estimate about 30 ATP are produced per glucose molecule. Well, the rest of them are going to come from the electron transport chain. So the electron transport chain comes at the end of this process of cellular respiration. Again, we've already talked about glycolysis, pyruvate processing, and the Krebs cycle, also known as the TCA or tricarboxylic acid cycle. And all of these processes need to go forward or occur before the electron transport chain can work properly, because the electron transport chain receives the protons that it needs from the production the production of the electron and proton carriers from these processes, specifically from the Krebs cycle, but also from the other processes. So as we've discussed before, the electron transport chain occurs in mitochondria in eukaryotes, and we need that NADH and the FADH2 to be delivered to the electron transport chain. And then ultimately, those electrons are going to be donated or received by oxygen, and that is the reason why we need oxygen to breathe, because that oxygen ultimately is going to be used to accept electrons at the end of the electron transport chain. That is why this form of producing ATP is called oxidative phosphorylation. Well, I feel like it needs more emphasis than that, right? So we're going to make it big. Oxidative phosphorylation, again, and that's opposed to or compared to substrate level phosphorylation, which is what occurs during glycolysis and the Krebs cycle or TCA cycle. So let's go back to the TCA cycle, and we see that we make NADH in a number of places. We also make FADH2. And each of these is, you could actually multiply by 2 because we have 2 molecules coming in here. Again, this is because in glycolysis, we split the original glucose molecule into 2. And so we have 2 molecules going through pyruvate processing, and we have 2 molecules going through the TCA, Krebs, whatever you want to call it. And here I just have depicted that all of these NADH and FADH2 molecules are going to go to the electron transport chain. Again, these NADHs and the FADH2s, they are not proteins. They are molecules that are really good at accepting electrons, and they can be bound by different types of proteins in the cell. And we know that because, so they're bound by the protein, the enzyme here, they're bound by the enzyme here, and then they could also be bound by proteins in the electron transport chain to then pass off the electrons there. So again, here's the summary. So you could come back and take a look at this slide for the electron transport chain. And here is a GIF. So this GIF, I think, really nicely illustrates the first step of the electron transport chain. And we start out with NADH. So first we'll focus on NADH, and then we'll talk about FADH2 later. But what happens with them is pretty similar. NADH comes in to the electron transport chain. So it is going to associate with an enzyme complex. It always associates with the same enzyme, which makes sense because we've learned that proteins are very specific in their chemistry and their shape to bind to specific molecules. And so this is always going to be the protein that NADH binds to. It's never going to come and bind to enzyme complex 2 or 3 because it just is not going to chemically or structurally fit together with them correctly. So what we see is that NADH comes and binds to enzyme complex 1. Enzyme complex 1 receives a hydrogen from NADH, and it actually splits up the hydrogen. It donates the electron to the next protein in the chain, in the electron transport chain, and then it actually pumps the proton across the membrane. And then we see that the electron is passed to complex 2, and there are other protons in the cell. As we know, the cell is mostly filled with water, and in a water solution, we often have protons because water can be broken up into H plus and OH minus. So then the electron can be passed on, and there is proton pumping going on in multiple enzyme complexes. All right, so what is actually happening here? You know, we could describe this, we could say there's the passing of electrons, but what does that really mean at the molecular level? It means that that electron is being bound by these proteins through interactions with their amino acids, and it causes a conformational change in the proteins. So we've talked about this a lot before. There's some kind of shape change. This shape change can do different things depending on what protein complex you're talking about, or enzyme complex. So in enzyme complex 1, it is going to pump a proton. So the conformational change, when it gets a hold of that hydrogen atom, the electron is going to associate with certain amino acids in that enzyme complex, and then the conformational change makes it the enzyme complex pass off that electron to this carrier that's labeled as Q right here. Then the other thing that occurs through that conformational change, it's the same shape change that caused the transfer of the electron to this carrier complex, that is going to pump the proton across the membrane. So next, the electron is going to be passed to enzyme complex 2. Sorry, this doesn't have a slider, so I can put it back and forth, but it goes to enzyme complex 2, and then it causes a conformational change again. Again, that is through the binding and the release of some of the energy from the electron. And again, that conformational change allows enzyme complex 2 to pass on the electron, and it also allows it to bind a proton and pump it through the membrane. And this is pumping, so we've talked about the difference between just allowing a molecule to move down its concentration gradient, but this is not that. This is pumping against the concentration gradient. So we get the proton moving across the membrane, and then the electron is passed on again to cytochrome C, and then passed to enzyme complex 3. The takeaway from this is that we are building up a proton gradient. So protons are going to be higher in concentration on one side of the membrane, and that whole process is fueled by the energy from this electron. And these molecules that we constructed in previous steps of cellular respiration are the ones that are donating that electron, so the NADH. And FADH2 also donates. It does not bind to enzyme complex 1. I think it actually binds to enzyme complex 2 instead. But the same thing essentially happens. We're donating electrons. These electrons are donating energy or releasing energy as they are bound by these proteins. And this causes both the transfer or the passing on of the electron and the pumping of protons. Not every single protein pumps protons. We saw two proteins here, the Q and the cytochrome C, which do not pump protons, and that's fine because other enzymes are. So an important question we need to ask here is whether all these proteins in the electron transport chain bind electrons equally well. And this is related to the question on the bottom here. Is there a specific sequence of proteins that the electron passes through? Why or why not? Now I'll take a moment to describe this diagram here. We have the proteins in the electron transport chain. The first one on the left is complex 1, which binds NADH and then releases NAD+. Then we see the second protein complex, the third, and the fourth, finally. And finally, also we see the half O2, which is going to be the final electron acceptor. It receives the electron after it's passed all the way through the electron transport chain. Now we said that the electron transport chain always binds to NADH with complex 1 because it's chemically and structurally compatible. However, if we could change that, could we have the electron kind of starting at complex 3 and then going to 2 and then to 1? And the answer is no, because these proteins have a different affinity for electrons. And I believe that there's a diagram related to this in your textbooks and in the reading that we did on NCBI, I decided to use this very straightforward diagram here. So we see that increasing affinity for electrons, it goes down this way, and we see the different complexes and the different proteins for the electron transport chain. So according to this graph, which is a little bit tricky because the highest electron affinity is at the bottom, not at the top, but we see what's at the bottom with the highest electron affinity, that's actually the half O2. So we are moving from molecules with a lower affinity for electrons to molecules with a higher affinity for electrons. So we could look at NADH and then complex 1, 2, 3, and 4. So as we move to subsequent proteins in the electron transport chain, we see that they have a greater affinity for electrons. And in addition, we could put an arrow going in the opposite directions to show that the energy left in the electron after it is received by each subsequent complex has decreased. So the electron has the most energy in NADH. Then a little bit of energy is released as the complex 1, the NADHQ oxidoreductase, uses some of that for a conformational change to pump a proton. And then complex 2 uses a little bit of the energy, so there's even less energy in the electron for complex 3, and so on. But these complexes have a high affinity for electrons, and so the electrons keep being passed between them. That idea is shown here in this nice little GIF, which I have a link to. And this shows the electron moving from complex 1 down the electron transport chain. And it shows that the energy of the electron goes down as it moves to subsequent complexes. The energy of the proton gradient increases because as we go through subsequent complexes, they are pumping subsequent protons. And we also now know that the affinity of each of these subsequent complexes is higher than the last for the electron. So we are increasing in affinity for an electron as we move forward here. So as we see, there is a specific sequence to the electrons being passed through the electron transport chain. We've seen why, and it is due to both the affinity for electrons of each of the individual proteins, as well as the individual proteins' ability to bind with one another, so they bind in sequence. And we will talk about the outcome of the proton gradient in the next video.